19. Genetic technology. Gene technology, also known as genetic engineering or genetic modification, provides ways to make changes to genes, the sets of instructions in the cells of all living creatures. There is a large amount of overlap between gene technology and the newer term synthetic biology. 19.1 Principles of genetic technology. Gene technology has been used to manipulate DNA to produce human proteins. This process is called genetic engineering. The mRNA that codes for the protein is identified and is extracted using high-density ultracentrifugate fusion. It is converted to copy, cDNA, using an enzyme called reverse transcriptase. It is converted to double-stranded DNA using DNA polymerase. Restriction endonuclease cut the DNA at specific base sequences and extract the gene with sticky ends. It is also used to cut open a vector, plasmid or virus, to ensure that complementary base pairs form. The gene is combined with the plasmid using an enzyme called DNA ligase. The vector is inserted into a host cell, usually E. coli, by using a cold 0.1 m CaCl2 solution. This causes the recombinant plasmids with inserted genes, to adhere to the bacterial cell walls. It is warmed to 37 C, which encourages the bacterial cells to take up the plasmids and the protein is produced. 19.1 to 1 Recombinant DNA. Recombinant DNA technology involves using enzymes and various laboratory techniques to manipulate and isolate DNA segments of interest. This method can be used to combine, or splice, DNA from different species or to create genes with new functions. The resulting copies are often referred to as recombinant DNA. Genetic technology involves using a variety of techniques to investigate the sequence of nucleotides in DNA and alter an organism's DNA. 19.1 to 2 Deliberate manipulation of genetic material. Genetic engineering, also called genetic modification, is a process that uses laboratory-based technologies to alter the DNA makeup of an organism. This may involve changing a single base pair, AT or CG, deleting a region of DNA or adding a new segment of DNA. Genetic engineering involves extracting DNA from one organism and placing it into the DNA of another to form recombinant DNA, rDNA. Genes must be inserted in such a way that they will be expressed in the genetically modified organism, GMO. 19.1 to 3 Explanation of genes to be transferred into an organism. Gene transfer, the introduction of new DNA into an existing organism's cell, usually by vectors such as plasmids and modified viruses. Cells may be modified ex vivo for subsequent administration to humans, or may be altered in vivo by gene therapy given directly to the subject. In order to transfer DNA into a living cell such as a bacterium, a vector, carrier, is needed. Common vectors are plasmids, circles of DNA found in bacteria, or viruses. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. 030046100457. 99. 19.1 to 4 The role of different enzymes. In molecular biology, endonucleases are enzymes that cleave the phosphodiester bond within a polynucleotide chain. Some, such as deoxyribonuclease I, cut DNA relatively non-specifically, while many, typically called restriction endonucleases or restriction enzymes, cleave only at very specific nucleotide sequences. DNA ligase is a type of enzyme that facilitates the joining of DNA strands together by catalyzing the formation of a phosphodiester bond. It plays a role in repairing single-strand breaks in duplex DNA in living organisms, but some forms may specifically repair double-strand breaks. A DNA polymerase is a member of a family of enzymes that catalyze the synthesis of DNA molecules from nucleoside triphosphates, the molecular precursors of DNA. These enzymes are essential for DNA replication and usually work in groups to create two identical DNA duplexes from a single original DNA duplex. Restriction endonucleases are enzymes that cut across DNA at specific sites, known as restriction sites. Restriction enzymes can make staggered cuts in DNA that gives rise to short lengths of unpaired bases known as sticky ends or straight cuts to give blunt ends. Pieces of DNA with sticky ends that are complementary to other are able to join together by forming hydrogen bonds. The enzyme DNA ligase joins together the sugar phosphate backbones of pieces of DNA by catalyzing the formation of phosphodiester bonds. In genetic engineering, vectors are used to carry pieces of DNA into cells. Typical examples are plasmids, viruses, and liposomes. Cells that have taken up plasmids with the desired gene can be identified by detecting fluorescence, GFP, or appropriate staining, GUS. Lengths of DNA for genetic modification can be synthesized directly from mRNA by using the enzyme reverse transcriptase. Specific lengths of DNA can also be synthesized from nucleotides using knowledge of the genetic code. 19.1-5 The need of promoter to be transferred. Plasmids are small circles of double-stranded DNA. They are useful for genetic engineering because they can be cut with restriction enzymes and have promoters and gene markers, e.g. genes for antibiotics, GFP or GUS, inserted into them alongside the genes transform the host cell. A promoter must be inserted alongside the gene because organisms will not transcribe and express a gene unless there is a binding site for RNA polymerase. 19.1-6 The confirmation of gene expression. Gene expression is the process by which the information encoded in a gene is turned into a function. This mostly occurs via the transcription of RNA molecules that code for proteins or non-coding RNA molecules that serve other functions. At present, the most accurate method of detecting gene expression is quantitative real-time PCR, RT-qPCR. With this method, reference genes that are stable in different biological systems and under different conditions can be obtained. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. 0300 461 0457. 100. 19.1-7 Gene editing. Genome editing, also called gene editing, is a group of technologies that give scientists the ability to change an organism's DNA. These technologies allow genetic material to be added, removed, or altered at particular locations in the genome. Several approaches to genome editing have been developed. CRISPR gene editing is a genetic engineering technique in molecular biology by which the genomes of living organisms may be modified. It is based on a simplified version of the bacterial CRISPR-Cas9 antiviral defense system. 19.1-8 Steps involved in the PCR. The polymerase chain reaction, PCR, is a method of making very large numbers of copies of DNA from very small quantities, even one molecule. In PCR, DNA is denatured by heat to separate the strands. A short length of DNA known as a primer attaches to one end of each strand so that DNA polymerase can start synthesizing a complementary strand using free nucleotides, as in replication. Heat-stable DNA polymerases are used in PCR. The first was Taq polymerase that is found in a thermophilic bacterium, Thermus aquaticus. A gene probe is a length of single-stranded DNA, which has a known base sequence and is used to hybridize with lengths of DNA which have the complementary sequence. Probes are labeled in some way to make them invisible, e.g. with radioactive phosphorus. PCR and gene probes are used in forensic investigations to look for matches between the DNA left at crime scenes and the DNA of suspects. 19.1-9 Use of electrophoresis. Electrophoresis is a laboratory technique used to separate DNA, RNA, or protein molecules based on their size and electrical charge. An electric current is used to move the molecules through a gel or other matrix. Electrophoresis is used to separate fragments of DNA of different lengths. The material to be tested is placed in wells in agarose gel and a voltage is applied across the gel. The 19th of January 10 The use of microarrays. Microarrays contain many thousands of genes and are used in two ways: to analyze the presence or absence of genes in different genomes and detect the presence of mRNA from cells to detect the genes that are being expressed at any one time. The 19th of January 11 Benefits of using database about nucleotide sequence. Databases hold huge amount of information on DNA and proteins. Bioinformatics deals with the storage and analysis of biological data, more specifically, nucleotide sequences and amino acid sequences of proteins. Information in databases is available to researchers. They can compare genes from different organisms, can search details of the various mutant alleles that cause genetic diseases, and compare the shapes of proteins. 19.2 Genetic technology applications in medicine. Genetic technology can provide benefits in, for example, agriculture and medicine, but has the associated risk of the escape of the gene concerned into organisms other than the. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. 030046100457. 101. Intended host. The risk is seen to be particularly high for genetically modified crops that are released into the environment to grown. The social implications of genetic technology are the beneficial effects of this technology on human societies. Ethics are sets of standards by which a particular group of people agree to regulate their behavior, distinguishing an acceptable from an unacceptable activity. Each group must decide, first, whether research into gene technology is acceptable, and then whether or not it is acceptable to adopt the successful technologies. Reproduction in plants. Sexual reproduction enables plants to pass their genes on, add to the number of the species, increase variation, and evolve. Flowering plants produce spores before they produce gametes. There are two types, microspores and megaspores. Microspores or pollen grains are transferred by wind or an animal vector to the stigma of another flower, often on different plant of the same species. The pollen grain grows into a tube that penetrates the female part of the flower. Sperm nuclei travel along this tube to meet the egg. Double fertilization occurs in plants. This leads to a zygote and a triploid cell. The zygote grows into an embryo. The triploid cell develops into a food store, the endosperm. The ovule develops into a seed, which protects the embryo for a while. The ovary develops into a fruit. There are a variety of means to promote cross-pollination. Many plants can self-fertilize, but many have strategies to make sure that they cross-pollinate. Many plants reproduce vegetatively, by asexual reproduction. Commercial plant growers can clone plants, and keep all desirable characteristics, by taking cuttings, grafting, or by micropropagation. Some applications of recombinant DNA technology that have benefited humans. Application of recombinant DNA technology. Explanation. Genetically engineered microorganisms' human genes can be inserted into bacteria, which are then grown in fermenters. The bacteria produce a human protein. Large amounts of insulin and human growth hormone can be produced cheaply in this way. Genetically modified plants' desirable genes can be transferred from an organism to a crop plant. Potato plants that are resistant to attack by a virus and maize, corn, plants that are resistant to drought have been produced in this way. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. 030046100457. 102. Genetically modified animals' human proteins, such as hemoglobin and blood clotting factors, are already produced in the milk of transgenic cows, goats, and sheep. Human gene therapy involves inserting a normal gene into an organism's body to correct a genetic disorder. Severe combined immune deficiency, SCID, leaves some people with almost no functioning immune system. Inserting copies of a gene coding for the enzyme adenosine deaminase, ADA, into the blood cells of sufferers has been used to cure the symptoms of SCID. Mapping human chromosomes, the Human Genome Project. Scientists in over 1,000 laboratories around the world are contributing to the Human Genome Project. They aim to create a map of all human chromosomes, identifying the precise location of every gene. This will help to develop new gene therapy treatments. 19.2 to 1 Recombinant human proteins. Many recombinant human proteins are now produced by GMOs, such as bacteria and yeasts. This makes available drugs, such as recombinant adenosine deaminase, ADA, to treat SCID and recombinant factor VIII to treat hemophilia. This production also secures the supply of others that were previously available from other sources, such as insulin. Genetically engineered insulin is identical to human insulin and causes no side effects compared to pig's insulin. The disadvantages of genetic engineering include concerns over its safety because new pathogens could arise. There are also ethical issues involved, e.g., whether it is right for people to determine the characteristics of a child. 19.2 Advantages of genetic screening: a sense of relief from uncertainty. A greater understanding of your health and your cancer risk. Information to help make informed medical and lifestyle decisions. Opportunity to help educate other family members about the potential risk. 19.2-3 Gene therapy. Basically, there are two types of gene therapy. 1. Somatic gene therapy. 2. Germline gene therapy. Somatic therapy usually occurs in the somatic cells of human body. Germline gene therapy occurs in the germline cells of the human body. Gene therapy is used in the replacement of genes that cause medical ill health. The method generally destroys the problem-causing genes. Gene therapy helps the body to fight against diseases by adding genes to the human body. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. 030046104457. 103. Gene therapy is employed to treat diseases such as cancer, ADA deficiency, cystic fibrosis, etc. 19.2-4 Ethical considerations about genetic screening and gene therapy. Concerns about the use of recombinant DNA technology. Moral or ethical concern explanation. Mutation of transgenic bacteria or viruses. If they mutate, these transgenic organisms could become new pathogens. The danger is that we might not be able to control these new pathogens. Genetically modified crops could escape. If pollen and seeds were carried from test plots, they might result in genetically modified populations elsewhere. Since the genetic modification could involve resistance to herbicides, these plants could become superweeds that we could not control. Transgenic organisms might set up an evolutionary process that could harm the environment. Some crop plants have been given a gene enabling them to produce a pesticide. Through natural selection, this might speed up the evolution of pesticide-resistant insects. Populations of transgenic organisms could upset the balance of nature. Populations of transgenic salmon have been produced in which individuals grow rapidly. These transgenic fish could compete for food with other fish species. The latter could become extinct and natural food webs could change. Objection to specific transgenic organisms. Many religious groups could not use products from specific organisms, e.g. to Hindus, cows are sacred animals, and to Jews and Muslims, pigs are unclean. The use of products from these organisms might be unacceptable to people from these religions. Eugenics. Genetic engineering could be used to insert genes into humans, or their eggs and. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. 030046104457. 104. Sperm. In some cases, for example SCID, this process could prevent an early death. However, it could be used to give individuals characteristics that are considered to be desirable. This is unacceptable to many people, and it also reminds people of some of the programs that have been used throughout modern history to eradicate less powerful ethnic groups. Screening could lead to discrimination against individuals. When fetuses are screened for genetic disorders, parents often face dilemma about aborting affected fetuses. It will become possible to screen adults for genes that predispose them to genetic disorders. This might lead to insurance companies discriminating against people with these disorders, even though they have perfect health. 19.3 Genetically modified organisms in agriculture. Genetic engineering is a technique used to deliberately modify a specific characteristic, or characteristics, of an organism. The technique involves removing a gene, or genes, with a desired characteristic from one organism and transferring the gene, using a vector, into another organism where the desired gene is then expressed. The genetically engineered organism will then contain recombinant DNA and will be a genetically modified organism, GMO, although plants and animals have been genetically engineered to produce proteins used in medicine. The main purpose for genetically engineering them is to meet the global demand for food. Crop plants have been genetically modified to be resistant to herbicides, increases productivity, yield. Resistant to pests, increases productivity, yield. Enriched in vitamins, increases the nutritional value. Farmed animals have been genetically modified to grow faster. It is rarer for animals to be modified for food production due to ethical concerns associated with this practice. Scientists have genetically modified many organisms including bacteria, for example, to produce insulin, sheep, for example, to produce a human blood protein known as AAT, maize, for example, to be resistant to insect attacks, rice, for example, to produce carotene to provide vitamin A. The benefits of using genetic engineering rather than the more traditional selective breeding techniques to solve the global demand for food are, organisms with the desired characteristics are produced more quickly. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. 030046104557. 105. All organisms will contain the desired characteristic. There is no chance that recessive allele may arise in the population. The desired characteristic may come from a different species, kingdom. 19.3 to 1 Role of genetic engineering in food production. There are many threats to the biodiversity of ecosystems. These include climate change, competition, especially with alien species, hunting by humans, and degradation of ecosystems leading to habitat loss. These factors are responsible for local extinction of populations and the total extinction of species. There are moral and ethical reasons for maintaining biodiversity, and also more practical ones. For example, plants may be sources of future medicines and animals may provide alleles to use in animal breeding. Genetic screening involves testing people to find out if they carry any faulty alleles for genes that can cause disease. There are genetic tests for many genetic diseases including breast cancer associated with BRCA1 and BRCA2, Huntington's disease and cystic fibrosis. Genetic counselors may help people who find that they or their unborn children have a disease-causing allele to make a decision about how to act on this information. Gene therapy involves the addition of genes to human or animal cells that can cure or reduce the symptoms of genetic diseases, such as SCID and some inherited eye diseases. Successful gene therapy involves selecting a suitable vector, such as viruses or liposomes, or inserting DNA directly into cells. Genetic engineering is used to improve the quality and yield of crop plants and livestock. Crops, such as maize and cotton, have been genetically modified for herbicide resistance and insect resistance to decrease losses and increase production. 19.3 to 2 Ethical and social implication of using GMOs. There are some social reactions towards GMOs, religious, social, medical etc. e.g. taste, unnatural organisms, allergies, biological warfare, fight against nature etc. Possibility to produce genetically engineered crops with novel properties. Examples of ways in which genetic modification could make crops more useful. Improve resistance to pests. Improve resistance to bacterial, fungal or viral diseases. Improve resistance to herbicides, so that plants are unaffected by herbicides used to control weeds. Remove allergens from crops that currently contain them, e.g. rice. Enable plants to resist cold or drought. Produce salt-tolerant varieties of crops, important in parts of the Indian subcontinent and sub-Saharan Africa if global warming causes a rise in sea level. Improve storage time, e.g. by slowing the sprouting of potatoes. Enable plants to produce pharmaceutical substances, such as edible vaccines. Enhance the production of vitamins and anti-cancer substances by plants. Advantages. Insulin produced was genetically identical and no side effects occurred. Insulin was originally extracted from animals, which was inhumane and its use conflicted with religious beliefs. Lecture notes for A-Level Biology by Mohammad Rashid Chatha. 030046104457. 106. Chymosin is produced from genetically modified yeast cells and coagulates casein in the dairy industry. Originally, renning was used, which was obtained from a calf's stomach, and vegetarians refused to eat dairy products that contained renning. Potentially, plants could be genetically modified to be resistant to diseases or have the gene for nitrogen fixation introduced. This would result in an increased crop yield, i.e. be more profitable. Production of human hormones, e.g. somatotropin, human growth hormone, was in short supply, but large amounts can be produced by genetic engineering. Gene therapy, viruses inserting a healthy gene into cells of individuals suffering from a genetic disorder, could help many people, e.g. those with cystic fibrosis. Disadvantages. Safety. Genetically modified organisms could have a catastrophic effect on other species, or new pathogens may arise. Ethical issues. Is it right to transfer genes between species, for companies to patent genes, for people to decide genes to determine the characteristics of a child, or alter genes in sperm and ova? Genetic engineers isolate, cut out and transfer genes between organisms. Genetic engineering technology has many applications, including making human products such as insulin on a large scale. The Human Genome Project has now produced the complete sequence of all 30,000 genes in the human genome. Knowing this sequence will provide a big boost for research in many areas of biology and medicine. Strands of DNA are cut using restriction endonuclease enzymes, which always cut DNA at a particular base sequence. Strands of DNA can be joined by using DNA ligase enzymes. Once the foreign DNA is inside the host cell, it is adopted by the host. Genes in the foreign DNA are expressed using the host cell's synthetic machinery, ribosomes, mRNA, etc. In addition, the foreign DNA is cloned, copied, every time the host cell divides. Gene therapy is a technique that replaces faulty genes with healthy ones. It provides great hope for sufferers of genetic diseases such as cystic fibrosis. The polymerase chain reaction, PCR, allows samples of DNA to be cloned in a test tube using the necessary enzymes, instead of inside a host cell. Many recent developments in this field have made cloning whole animals and organs possible. However, although the technical problems have been overcome, the ethical difficulties still pose a great problem for this area of research. Ecology. Types of interactions that occur between organisms in an ecosystem. Activity. Type of interaction. What happens? Reproduction. Intraspecific location of selection and competition for a mate. Caring for young. Intraspecific. Both parents, one parent or, more rarely, older siblings feed the young, keep them warm and sheltered and protect them from predators. Social behavior. Intraspecific animals cooperate to find food and to defend. Lecture notes for A-Level Biology by Mohammad Rashid Chatha. 030046104457. 107. Themselves against competitors or predators. Competition. Intraspecific organisms compete for resources such as food, space, light, water and mineral nutrients. Reproduction. Interspecific animal vectors pollinate some species of plant and help to disperse the fruits and seeds of others. Caring for young. Interspecific. Rare, but some species rear the young of another species. The cuckoo, for example, lays its eggs in the nests of smaller birds that then rear the fledglings. Mutualism. Interspecific. Two organisms both benefit from a long-term association. Parasitism: interspecific individuals from one species use another species to provide food and shelter while giving nothing in return. Many hosts actually suffer from the interaction. Many parasites cause disease. Predation: interspecific: all animals obtain food by eating another organism. In its widest sense, predation also applies to herbivores feeding on plants. Protection: interspecific: many species make use of other species to hide from predators. Some copy the markings of other species to mimic warming signals that predators avoid. Competition: interspecific organisms in a community compete for resources such as food, space, light, water, and mineral nutrients. Defense: interspecific organisms have developed various strategies to defend themselves against predators, parasites, and competitors, including mimicry. An ecosystem is made up of abiotic, or physical, components and biotic, or living, components. Abiotic factors that affect an ecosystem include climate and weather, including humidity, temperature, air and water movements, soil type, rock type, natural catastrophes, water and mineral availability, and the position of the ecosystem on Earth. Soil type helps to determine the type of plant that grows in an ecosystem. This affects which herbivores can live there and, in turn, which carnivores live there. The size of rock particles and the amount of humus present determines soil type. There are three main soil types: clay, silt, loam, and sand. Loams are the most fertile soils because they contain a balance of particles and are rich in humus. Temperature, light availability, humidity, water availability, and wind and water movement are abiotic factors that you need to study in more detail. You can measure them in the field using simple techniques. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. 030046104457. 108. Biotic factors describe the interactions between living organisms. Interspecific interactions occur between organisms of different species. Intraspecific interactions occur between organisms of the same species. Biotic and abiotic factors affect each other. The physical environment determines the distribution of organisms in the ecosystem, but living organisms can also change the physical properties of an ecosystem. Energy transfer through organisms in an ecosystem is one way. Elements such as carbon and nitrogen cycle between living organisms and the abiotic environment. In a food chain, energy is passed from producers to primary consumers to secondary and tertiary consumers. Each level of the food chain is called a trophic level. In most ecosystems, many food chains are linked together in a food web. Decomposers break down the dead bodies of producers and consumers and play an important role in mineral cycling. Most decomposers are bacteria and fungi. There are three main types of numbers: pyramids of biomass and pyramids of energy. Pyramids of energy show that over 90% of the energy transferred to the next trophic level is lost as heat. The carbon and nitrogen cycles show how these elements circulate between the biotic and abiotic parts of an ecosystem. You should appreciate the overall cycle and be able to draw a clear summary diagram. Carbon dioxide is greenhouse gas. Together with other gases in the atmosphere, it acts as a heat insulator. The presence of CO2 in the atmosphere helps to maintain temperatures on Earth that are warm enough to sustain life. Human activities have led to an increased concentration of CO2 in the atmosphere. Many scientists believe that this is causing a general increase in average temperatures throughout the world, a phenomenon commonly called global warming. Nutrition in man. The main functions of the liver. The thousands of different chemical functions performed by the liver contribute greatly to the overall composition of the blood. They can be grouped under a few basic headings. Control blood glucose level, control of amino acid levels, synthesis of plasma proteins, synthesis of fetal red blood cells, destruction of red blood cells, detoxification, production of bile, control of lipid levels, storage of vitamins, cholesterol formation. Human activity and the environment. Humans influence the environment of the Earth on a global as well as a local level. Farming is the major human activity that causes environmental danger. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. 030046104457. 109. People need to farm to feed the increasing human population, but many farming techniques cause long-term damage to the land, lowering its capacity to grow food in the future. To avoid this problem, it is important that farmers start to use sustainable methods of agriculture. Many human activities cause pollution. Pollutants can affect the air, the sea, and rivers and lakes, and the land. Acid rain, use of CFCs and other substances that destroy the ozone layer, and increased production of greenhouse gases all contribute to air pollution. Water can be contaminated by organic pollution. This can lead to a sequence of events called eutrophication. Heat, oil, and other substances can also pollute waterways. When forests are cleared to make new farmland, fewer trees are available to photosynthesize, we lose species diversity, and the land becomes vulnerable to erosion. Severe soil erosion can lead to desertification. Many sustainable methods of agriculture try to minimize the effects of soil erosion. Practical skills for A-Level. A hypothesis about the relationship between two variables predicts how one variable affects the other. It should be testable and falsifiable by experiment. To make up a 1% mass volume solution, dissolve 1 g of the solute in a small amount of water, then make up to a total volume of 1 dm3. To make up a 1 mol dm3 solution, dissolve 1 mole of the solute in a small amount of water, then make up to a total volume of 1 dm3. The mean of a set of data is calculated by adding up all the individual values and dividing by the total number of readings. There is a 95% probability that the true mean lies within, plus 2 standard errors of the mean you have calculated. This can be shown by drawing error bars on a bar chart, where the error bar extends 2 standard errors above and below the plotted value. If the error bars for 2 sets of data overlap, then there is no significant difference between significant difference between the two sets of data. If the error bars do not overlap, it is possible that there is a significant difference between them, but this is not necessarily so. Keywords: bioinformatics, Bt maze, cystic fibrosis, DNA ligase, DNA polymerase, embryo biopsy, gel electrophoresis, gene editing, gene therapy, genetically modified organism, GMO, genetic engineering, genetic screening, genome sequencing, GM salmon, golden rice, hemophilia, Huntington's disease, ligases, liposome, marker gene, microarrays, plasmids, polymerase chain reaction, PCR, promoter sequence, recombinant DNA, recombinant protein, restriction endonuclease, reverse transcriptase, severe combined immunodeficiency, SCID, sickle cell anemia, Taq polymerase, vector. Chapter Bioinformatics: the development of the computer tools and software required to organize and analyze unprocessed biological data. Bt maze is a variant of maize genetically engineered to produce its own insecticide, the Bt toxin, making it resistant to corn borers. Cystic fibrosis is an autosomal recessive condition characterized by the production of thick, sticky mucus that causes lung infections and scarring. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. 0300 4610457. 110. DNA ligase is an enzyme that joins the sugar phosphate backbone of two DNA segments. DNA polymerase is an enzyme that synthesizes a double-stranded molecule of DNA from a single template strand using complementary nucleotides. Embryo biopsy is a few cells from an embryo can be extracted and screened for genetic diseases. Gel electrophoresis is a type of chromatography that separates nucleic acid fragments or proteins by size using electric current. Gene editing is a type of genetic engineering that involves the deletion, insertion, or replacement of DNA at particular sites in the genome. Gene therapy is a therapeutic technique in which a faulty allele is replaced with a functional allele in order to treat or prevent disease. Genetically modified organism, GMO, is an organism that has had its genome altered. Genetic engineering is the modification of the genome of an organism by the insertion of a desired gene from another organism. This enables the formation of organisms with beneficial characteristics. Genetic screening is the testing individuals for specific genes or faulty alleles. This can be used to detect disorders, e.g. hemophilia, sickle cell disease, cystic fibrosis, Huntington's disease, or to identify an increased risk of some cancers, e.g. breast cancer. Genome sequencing is determining the entire DNA nucleotide-based sequence of an organism. GM salmon is genetically modified salmon. A gene that is inserted into fertilized eggs results in an increased appetite, enabling the fish to reach the desired market size rapidly. Golden rice is a variety of rice genetically engineered to produce beta-carotene, a precursor of vitamin A. Hemophilia is a sex-linked genetic condition caused by a mutation of the factor VIII, F8, gene on the X chromosome. This disrupts the body's ability to form clots. Huntington's disease is a genetic disorder characterized by the progressive degeneration of nerve cells in the brain, reducing the affected individual's ability to think, talk and move. Ligases is enzymes which catalyze the joining of two molecules, usually via a hydrolysis reaction. Liposome is a vesicle with a lipid bilayer that can be used to administer medication. Marker gene is an additional gene inserted into a plasmid that is used to aid in the identification of host cells that have taken up the desired gene. Marker genes are easily recognizable, e.g. fluoresce, provide antibiotic resistance. Microarrays is a method of DNA sequencing. The DNA sample hybridizes with fragments of single-stranded DNA bound to a chip. Plasmids are loops of DNA found in the cytoplasm of prokaryotic cells. They can be used as vectors in genetic engineering. Polymerase chain reaction, PCR, is an in vitro technique used to rapidly amplify fragments of DNA. Promoter sequence is a region upstream of a gene where the transcription initiation complex is formed. Recombinant DNA is a combination of DNA from two different organisms. Recombinant protein is a protein produced as a result of the expression of recombinant DNA in a cell. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. 030046104557. 111. Restriction endonuclease are enzymes that cut DNA molecules at recognition sequences, creating sticky ends. Reverse transcriptase is a viral enzyme which produces cDNA from an RNA template. Severe combined immunodeficiency, SCID, is a group of polygenic diseases which disrupt the development of B and T lymphocytes, resulting in persistent infection. Sickle cell anemia is a disease characterized by hook-shaped red blood cells. A missense point mutation of GAG to GTG in the HbA, normal, allele for the globin polypeptide results in the HbS, sickle cell, allele of the gene. Taq polymerase is a DNA polymerase from a bacterial species which is adapted to live in extreme heat. It is used in PCR technology to produce fragments. Vector is a carrier used to transfer a gene from one organism to another, e.g. plasmid, virus, liposome.